everybody, welcome back to Teach Me to Science. My name is Saren and today I'm going to be talking about PCR. First I'll define what PCR is and talk about who discovered it. Then we'll talk about how PCR works and why it's helpful to scientists. PCR stands for Polymerase Chain Reaction. PCR is a laboratory technique which is used to make many copies of DNA or a section of DNA called a gene. This process creates exponentially more copies of the DNA or gene you're interested in. The process of PCR was discovered by Cary B. Mullis. He created the very first version of this technique in 1985. Nowadays, the process of PCR is used widely around the world in laboratories all over. But when the technique was first created, it was very long and tedious. Now we have PCR machines called thermocyclers, which automatically run PCR for us. Now let's talk about the basic process of PCR. PCR begins with a template strand. This is the strand of DNA or gene that you're interested in replicating. In order to successfully replicate a section of DNA, you have to break the two strands of DNA apart. This can be done by breaking the hydrogen bonds that connect the strands of DNA. Next, you have to add in the primers. Primers provide a starting point for the machinery to replicate your section of DNA. Without a primer, your replication machinery will be lost and won't know where to start replicating your DNA. After the primers have bound to their locations on the DNA, the replication machinery will then bind and replicate your strand of DNA. So how would you perform PCR in a lab? First, you would add all of the different components to a single PCR tube. This includes the template strand, primers, nucleotides, which are the building blocks of DNA, and the DNA replication machinery. Try and think about what would happen if each individual component was left out of this mixture. This tube with all of the components then undergoes thermocycling. Thermocycling is a series of increasing and decreasing temperatures over a period of time that actually does the replicating of your DNA. Without changes in temperature, no DNA will be replicated. Here's a diagram that shows at what point of the thermocycling process each event happens. The initial temperature increase breaks the two strands apart. A slight decrease in temperature then allows for the primers to bind to each strand. And finally, a slight increase in temperature causes the replication machinery to replicate the section of DNA. Each of these steps has a specific name, called denaturing, annealing, or extending. Denaturing is the process of separating the two strands of DNA. Annealing is the step where the primers bind to each strand. And extending or extension is the process where the machinery replicates the strand. So why is the process of PCR so helpful to scientists? PCR is helpful when our samples are too small to work with, or we need more of a specific segment of DNA. It's also very helpful for forensic sciences. That's all I have for today's video on PCR. If you could, please like this video and subscribe to my channel. I would really appreciate it. Thanks. Bye!